Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. In this class, we shall discuss a very important tool used in biotechnology, that is PCR. PCR is also called polymerase chain reaction. So here, whenever there is a amplification of DNA, you might have heard the term amplification. Amplification means to maximize. That means we heard of sound amplification. That means the little sound will be heard bigger. So here amplification refers to, it is the process of multiplication or obtaining the multiple copies of DNA from a small amount of DNA. That is called amplification. That is synthesis of multiple copies of DNA from a small amount. So that is called DNA or gene amplification. So this amplification process was made possible with the discovery of polymerase chain reaction by Carey B. Mullis in the year 1980s. From there was the acceleration given to the biotechnological works. You may ask the question now, why there is a need of amplification of DNA? Suppose if there is any crime happened, okay, from that place, for DNA fingerprinting, they'll collect the samples like hair, blood. Okay, suppose, consider they collected blood. So from that blood, the DNA will be isolated, and make sure that the DNA can be isolated only from WBCs. As we know, only WBC contain nucleus, not the RBCs. So from that, they obtain a DNA of very little amount. So from that, they will conduct the DNA fingerprinting technique. If anything goes wrong, then there is a need of again the repetition. The process should be repeated again. So if the amount of DNA is very little and they have to conduct the procedure once again. So at that time, what they will do? So they will go for a technique of PCR, from which a little amount of DNA can be obtained in a bulk amount, or the DNA can be obtained in a multiple copies, and that was made possible with the discovery of PCR. Not only that, if any biotechnological works or gene cloning procedure is conducted, suppose they isolated the gene coding for insulin from human cell. So they'll isolate once and they will clone for the bacteria. If that something goes wrong and there was no successful transformation, then again they have to go for the procedure. Again, instead of starting from the isolation, the things like that, if the gene obtained is amplified in PCR, then the further process will be made easy. Okay. Now, this PCR technique conducted in a machine, we call it as PCR machine. Or this PCR machine is also called thermal cycler. Thermal cycler, because there is a cyclic repetition of different temperatures. For example, DNA amplification in PCR will takes place in 3 steps like denaturation, annealing and primer extension. And for these 3 steps, it is in need of 3 different temperatures. So that temperature will not be controlled manually, but it is controlled auto by PCR machine. That's why it is called thermal cycler. It on its own will control the temperature required for different steps in PCR. And the requirements for PCR, the basic requirements for PCR. The primary requirement is the genomic DNA to be amplified. Genomic DNA to be amplified. And preferably your double-stranded DNA will be taken. Then the second one is PCR machine, or we call it as thermal cycler. Then DNA polymerase. You may ask a question. DNA polymerase is available in every organisms including human beings. Okay, so that DNA polymerase cannot be used in PCR because in PCR there is a maintenance of high temperature up to 94 degrees Celsius, and that high temperature cannot be withstood by the DNA polymerase which is present in the organisms like human beings or normal bacteria. So for that, a special DNA polymerase is used, that is called Taq polymerase. Taq polymerase or Taq DNA polymerase, we can refer it. This speciality of this DNA Taq polymerase is, this Taq polymerase is isolated from a bacteria called Thermus aquaticus. And Thermus aquaticus is a thermostable bacteria. It is present in very high temperatures. Since it is adapted for very high temperature, the enzymes present in it, including DNA polymerase, can withstand very high temperature. For that, they isolated Taq polymerase from Thermus aquaticus and it is applied in PCR. Then the next requirement is I'll write here. Fourth requirement is for polymerization of DNA. You have studied in replication of DNA. During replication, there is a synthesis of RNA primer which will give the OH, 3' OH for the polymerization of nucleotides. Likewise, here also there is a production of multiple copies of DNA, polymerization taking place. For polymerization, there is always be a need of 3' OH group. So that 3' OH group is provided in the form of oligonucleotide primers. Oligonucleotide primers, and they are artificially synthesized. Okay, they are artificially synthesized DNA strands of about length 13 to 17 nucleotides, and they are complementary to, the ends of genomic DNA. So Suppose this is our genomic DNA to be amplified. This is 3 prime, 5 prime, 3 prime, 3 prime and 5 prime. So this is the DNA to be amplified. Say for example, the DNA primer is synthesized in such a way that it should be complementary to this 3 prime end. So here is a primer synthesis and this polarity will be 5 prime to 3 prime. Here is the 3 prime OH group. So here the polymerization will take place. And other primer will be here 5 prime to 3 prime. So these two are the oligonucleotide primers which are synthesized artificially and they will be complementary to the ends of single strand, the DNA which is to be amplified. Okay, then the fifth requirement of PCR is DNTPs. In DNA, there are 4 types of nucleotides present. So, for polymerization, 4 types of nucleotides should be provided and that will be provided in the form of deoxynitrogen triphosphates. In the form of triphosphates will be given. So, such as DATP, DGTP, CTP and TTP. So 4 types of nucleotides are added in many number. That depends on how much amount of DNA we need. Based on that, the number will vary. Types will be 4, but the number will vary depending upon the amount of DNA to be obtained. Okay, and the primers also. The number, 2 types of primers will be added, but the number depends upon the number of copies we want. Okay, then the 6th requirement, Mg2 plus magnesium ions, and it will be provided in the form of magnesium chloride. And the necessity of this is magnesium ions will act as cofactor for synthesis of cofactor for the enzyme DNA polymerase. For any enzyme to work, the catalyst is required. So that catalyst will be available in the form of cofactor Mg2 plus for DNA polymerase. When this cofactor binds to DNA polymerase, here we are using Taq polymerase, and that activity will be enhanced. So to enhance the activity of Taq polymerase, they make use of Mg2 plus ions, and that will be provided in the form of magnesium chloride. So all of these will be added to a tubes called, generally they are made up of plastic tube. A pair of tubes are available. So they will be having cone shape like this. To this a pair of tubes all the contain first the DNA to be amplified, then DNTPs, then Taq polymerase, then ions, magnesium ions, and the primers, oligonucleotide primers of 2 types will be added, and it will be placed in a thermal cycle. Okay, so here it will be placed in a thermal cycler, and everything will be auto-regulated here. That is, once the process is switched on, that is the PCR machine is switched on, everything will take place on its own. The only controlling is amount we want. So based on that, everything will be set and the machine will be on. So when the machine is on, the DNA synthesis will take place in 3 steps, such as first one, denaturation, then annealing, annealing, and trimer extension. Extension, or we call it as polymerization. Primer extension or polymerization. So let us discuss now all of these three steps in detail. There are three steps in PCR. The first one is denaturation. Denaturation, where the temperature of the thermal cycler during denaturation will be brought to 94 degrees Celsius. The temperature of cycler will be brought to 94 degrees Celsius. At this temperature, DNA will undergo denaturation. Okay, that means suppose this is the DNA which is to be amplified. Consider. So this is 5' end, 3', 3', 5'. I'll write some nucleotide sequence here so that it will be easier for you. So this is the sequence, and complementary sequence will be TA, C, GG, A, TT, C, GG will be the sequence, and this is the DNA to be amplified. So by the end of three steps, we want the same type of DNA in multiple copies. Let us understand now how it will happen. So now, during denaturation, the temperature will be brought to 94 degrees Celsius, right? So at this temperature, the bonds present between the nucleotides, will break. The hydrogen bonds will break, and this will be converted into two single strands. Okay, so like this. This will be brought to two single strands. So the sequence will be AT, G, CC, T, AA, G, CC will be the sequence. One strand, and the other strand T, A, C, GG, A, TT, C, GG. So this is the two strands which are separated. Okay, this will takes the time depends upon the length of DNA fragment or the amount of DNA fragments, but approximately we can take the time of 2 minutes. Let us consider. This takes of about approximately 2 minutes, and remember this time is not fixed. So that will vary depending upon the amount of DNA to be amplified and the length of DNA fragments. Then, after denaturation, the second step will be annealing. Annealing. So, annealing is the attachment of primer to 3' end of single strands. 3' ends of single strands. To the single strands, to the 3' ends, there will be a addition of, or there will be a bonding of primers like this. So this is a primer. Say for example, this is a primer. So it will be attached like 5' to 3' direction. And I already told they will be complementary to the 3' end of a single strand. So the sequence will be like this: A, T, G. So the length will varies. Okay. So multiple length you can write here. C, C. So here it will provide a 3' end. And here also the sequence will be G, G, C, T. So here is 5' and 3'. here it will provides the 3' end for polymerization. Okay, so that is called annealing, attachment of the primers to 3' end of single strands. Then after attachment, the polymerization or is also called primer extension. Polymerization or primer extension. So here in the content we have added already the deoxynitrogen phosphates, that is DNTPs we added. So those DNTPs will be added on the complementary basis to the 3' end of primer, and this process is carried out by the enzyme Taq polymerase. That means Taq polymerase will go on adding the nucleotides on a complementary basis. It is exactly like how the replication is taking place. So by the end of this polymerization, there is a synthesis of 2 copies of DNA. That is like, so here if you write this polymerization, so here it will be the polymerized strand, and this is from 5' to 3', and here also 5' to 3'. So the bonds will be formed on a complementary basis. So I write the same here. So here this is one strand, 5' to 3', 3' to 5'. So this will be exactly similar to this. So there is synthesis of 2 copies of DNA which is similar to this. Okay, so that is by the end of one cycle. So that is, let us take annealing, let us say it is completed in 2 minutes, and polymerization is completed in 3 minutes. Totally in 5 minutes we could obtain the 2 copies of double strands. Okay, now let us understand, and remember this timings will vary, so we cannot define the timings here, but we can define the copies of DNA. Suppose if this is the genomic DNA to be amplified, by one cycle, after completion of one cycle, we can obtain 2 copies of DNA. Okay, we took 1 copy of DNA and we could obtain 2 copies by the end of one cycle. And you only say what is the number of copies after second cycle. This is second cycle. Just think there are 4 single strands, right? 4 single strand means 4 complementary synthesis. So we will obtain 4 copies of DNA. And likewise, by the end of 3 cycles, we will obtain 8 copies. And by the end of 10 cycles, we could obtain 1024 copies of DNA. So you can calculate further also. So this is how the high content of DNA will be obtained in a short period of time with the usage of PCR. Okay, so these are all the tools used in recombinant DNA technology, and in the next class we shall discuss how the process of the recombinant DNA technology is conducted step by step. Okay.